Okay, in this video, we're going to figure out, once you know the amino acid composition and maybe the first amino acid, how do you actually sequence the rest of the protein? And this process is called Edman degradation. This is sort of the original way we did this. And people still do Edman degradation, although a lot of people will do high-resolution mass spec as well. But we're only going to focus on the Edman degradation right now. So in Edman degradation, the way it works is you label and remove the N-terminal residue. So you label and remove the N-terminal residue. So remember, N-terminal means the very first one. So you label, you remove it, and you identify it one at a time. And then you basically repeat the process. The labeling agent that you use is called Edman's reagent. And it's PITC, which stands for phenyl isothiocyanate. I should have gotten a regular size iPad instead of this iPad mini if I was going to be writing all these chem words. And the process looks something like this. So here is our PITC reagent with its thiocyanate group coming off of it. And here is the protein that we're interested in sequencing. So I'll just show it with a couple of amino acids here so you get the idea of how this is going to work. So here's R1, the first amino acid. Here's R. So again, this is going to be a reaction with the free amine right here at the carbon of the thiocyanate group. And what we're going to get is a derivative molecule. And that derivative will look like this. Might as well get a new page so I have lots of room. So here it is. And then I'll circle the amino acid so you can see it a little bit better. So right here now is our amino acid that we attached, and this is called the PTH derivative. So we've basically derivatized our amino acid derivative. And then we'll basically identify this by HPLC. And so the way this works is that you label it, you remove it, and then you run it over the HPLC and identify it. And then you repeat the process. And typically in a sequenator, which is an instrument that will do this for you so that you don't have to do it by hand, is they have a glass slide that has the peptide attached at one end. And then basically the reagents, the PITC and all the other solvents that are used for this chemistry, are added as a vapor across that glass slide. And so you can cut off one amino acid at a time, and the peptide is staying immobilized so you can work your way methodically down the chain. And this is so sensitive on this glass slide, it goes down to picomoles in terms of detection. However, like with any chemistry, you have to keep in mind that the more steps you have in like a synthetic reaction, the more your yield goes down. So the Edman degradation process works good for about 50 amino acids in a row, and then you start getting yield issues because you're just losing yield every single time that you go. So that means that if you want to do a larger protein, then you've got to sequence the protein in smaller bits. Okay, so if you want to do a larger protein, oh, I'm writing in red again. You've got to break, first of all, any disulfide bonds. That might be between cysteine amino acids. And those cysteine amino acids forming those disulfides could be intramolecular, so cysteines within one protein, or the protein that you're studying might have multiple bits that are not connected covalently, so other than through disulfide bonds. So you could have intra or intermolecular disulfide bonds. So that's the first thing you do. The second thing you do is you use enzymes, which are proteins that can specifically hydrolyze peptide bonds. So you use enzymes to break the protein apart in specific locations. This says specifically. And then you can sequence the smaller bits that you get and overlap those fragments to put together a complete protein sequence. So let's look a little bit at some of these steps. So let's look about how you break the disulfide bonds. So typically the way we do that is we add a reducing agent, right? Because disulfide bonds are oxidized thiol groups that are oxidized together. So we add a reducing agent, and typically that reducing agent is dithiothreotol, so DTT, dithiothreotol. And what that does is that takes our cysteines, so here's two cysteines with their thiol groups disulfide bonded. We add DTT, and now we get two cysteines with free thiol groups on the end. Now the important part about this is if you're doing this on the lab bench, it's an oxidizing environment, so you can get reoxidation in the absence of that DTT. So after you reduce those, we typically add iodoacetate. Iodoacetate, that's horrible. Iodoacetate, which will come in then and block that free sulfur group. So now you have cysteine that has the free thiol, and in the presence of the iodoacetate, which looks like this, you end up with cysteine with the acetyl group on it. So now you don't have to worry about those disulfide bonds reforming. And we'll see some examples later as we do some practice problems where you can do these experiments then where you use the proteases or the enzymes that break the peptide bonds in the presence and the absence of reducing agent to break those disulfides, and you get different results that are important for disulfide bond mapping. So in the next video, I'm going to do a sample problem, and it's good to know about table 3-6 on page 100 of your textbook, because this lists the various proteases. So these are the enzymes that specifically cut the protein at different locations. So for example, it shows trypsin, which is an enzyme, and it says that its cleavage points are lysine residues and arginine residues, and then it has a C, which stands for C-terminal domain. So that means that any time the trypsin encounters a lysine residue in the protein sequence, it will break the peptide bond immediately following that particular lysine, so on the C-terminal side. Whereas something like asp-N protease, if you look in table 3-6, looks like it cuts on the N-terminal side of aspartate. So anytime it sees an aspartate residue, it will cut before it. So you have to pay particular close attention to whether or not your protease is cutting after an amino acid or before it. But table 3-6 will give you a nice list. So again, you break the disulfide bonds, or in some cases you're going to do these experiments without breaking them. You cut them specifically with a particular protease, separate the fragments out by like SDS page, and then sequence each fragment by Edman degradation. And then you start the process over with a fresh protein and a different protease. And in the end, you get lots of data, and you can start overlapping fragments that you see in common to come up with the full sequence of the protein. It's like a puzzle.